Welcome to week 5. So, in this week's lecture, we are going to look at the unit of life, which is cell. So, you have already seen this, but this is a good time to recapitulate this. So, the levels of organization of life forms. The first or the most bigger level will be that of a biosphere, which includes all living entities of the universe, or if in this case we will consider it as the earth. So, our earth considers the biosphere, which accounts for all the organisms on this planet. The next level of organization is an ecosystem, which consists of an interacting community of different life forms. So, a very good example will be a rainforest, and in this rainforest we have individual organisms which are life forms or species such as plants, insects, birds, animals and microbes. If we consider a multicellular organism like us, like humans, then they consist of organs. So, we have all sorts of organs, and these organs have specialized functions. Examples will be your heart, lungs, eyes, right. Now, each of these organs are made up of tissues. So, these tissues again perform certain specialized functions. So, if we consider the organ eye, it will have 3 different types of tissues. One will be light sensitive tissue, another will be support tissue, and the third one is the retina, right. Now, if we zoom into each of these type of tissues, then they are made up of cells, which constitute the smallest unit of life. Microbes, they are unicellular, so they are a single cell organism. But if we talk about animals, birds, insects, plants, these are all multicellular. They will have these organs and tissues, and for them also the smallest unit of life will be a cell. If we consider eye, then together for all these tissues, if we look at all the different types of cells, it turns out that there are 58 different cell types, which can be grouped into these 6 different types such as photoreceptor, horizontal, bipolar cells, amacrine cells, retinal ganglion and non-neuronal cells. So, there is a very interesting project that is going on called the Human Cell Atlas, which aims to map all the different cell types that are present in a human being, right. So, this particular data is taken from this paper, where they mapped the different cell types in a human eye, and it turns out that there are 58 different cell types that are present. So, if we zoom in further, then for eukaryotic cells, we will see that they are made up of different organelles. So, these are distinct structures within the cell, and they perform different functions. We will see those in more details in today's lecture. If we zoom in further, so examples of organelles will be nucleus, mitochondria, etc. If we zoom in further, then these are made up of molecules. We have already seen these molecules: nucleic acid, protein, lipid, carbohydrate, etc. right. So, in the first 4 weeks, what we have learned was mostly focusing on these molecules, and these are called the molecules of life. And as you may have seen that these molecules are very common in all the life forms, whether it is bacteria or whether it is us, right. So, we have the same type of DNA, proteins are made up of the same type of amino acids, so on and so forth. Now, from this week onwards, we are going to look at these higher levels of organization. So, week 5 will focus mostly on cells, which is the smallest unit of life, and then from week 6 onwards, we will look at these higher order organizations, right. So, let us look at these various life forms that are present on this biosphere, which is Earth. If we look at all the life forms, they can be grouped into these 3 domains. So, these are called the 3 domains of life. First one is called bacteria, the second one is archaea, and the third one is eukaryotes. Bacteria and archaea, these are unicellular organisms, and they evolved much longer before the eukaryotes evolved. So, I will discuss about evolution of life in more details in another week's lecture. So, briefly, bacteria and archaea both are unicellular, and together they are called prokaryotes. Eukaryotes can also be unicellular, but they can also be multicellular. So, all plants and animals, they are eukaryotes. So, let us look at the scale of things, right, in terms of the dimension. So, if you look at your thumb, so it is just shown here, we are looking at something in the scale of centimeters. So, this is 20 millimeter, which is 2 centimeters. If we zoom in to a particular part of your thumb 10 times, then this is 2 millimeter, and we can see all these ridges. So, these ridges are the ones which actually form the fingerprint of your finger. If we zoom in 10-fold further, then you can start seeing these small structures which are cells. We zoom in 10 times further, now you can actually see the outline of a cell. So, this is 20 micrometer across. So, you can see from this that these cells are roughly 10 micrometer in size. So, this end to this end will be around 10 micrometers or 15 micrometers. And here we can see all these different organelles. So, two big prominent organelles can be seen here. So, this is the nucleus and that is the mitochondria. So, if we zoom in 10 times further, we can see mitochondria, and we will see that mitochondria is something that is very important. We will discuss more about mitochondria when I talk about respiration. So, you can see there are all these small dots which are floating around. So, if we zoom in 10-fold further, you can see that those dots are ribosomes. So, on those ribosomes, so this is the mRNA, and on that protein is getting synthesized. We zoom in further, we can see more details of the ribosome. We zoom in further, we can start seeing the atoms, and this is 2 angstrom, 0.2 nanometer or 2 angstrom, we can actually see the atoms, right. So, there are all these different techniques which can be used to see at this different scale length. So, I will talk about these techniques in more details in the next lecture. So, it turns out that cells are very small, they are in the range of 10 micrometer size, but that is a eukaryotic cell. If you think about bacterial cell, they will be even smaller. So, you may ask that why cells are so small? Why do we have to make the cells so small? Because we cannot even see it by naked eye. One of the explanation is that by making something smaller, we can increase the surface area. So, take for example, this cube. So, let us say it is 5 by 5 by 5 in some units, right. The volume of this cube will be 5 cube. So, 5 times 5 times 5, that is 125, and the surface area will be, so one of this face will be 5 times 5, that is 25. Now, there are 6 faces, so 6 times 25. So, if we divide that, we get 1.2. Now, instead of having one large cube, what we can do is we can divide this into multiple cubes, where each cube is unit 1 in length, breadth and height. The volume of this cube will be 1, and the surface area will be, so one of this area will be 1 times 1, so that is 1, and then there are 6 faces, so it will be 6. So, 6 by 5 is 6, right, and then you have the same 125 number of cubes. So, you multiply and divide by 125, so it remains as 6. So, for this, the surface area to volume becomes 6, right. So, this is 5 times of this. For the same material, we have increased the surface area to the volume ratio by 5-fold, right. And it turns out that surface area or the membrane of the cell plays a very important role. So, we will see that in the following lectures, and we have sort of seen that when I discussed about membrane proteins, right. So, these membrane proteins are embedded on the membrane and they do their function. So, you need a large surface area, right. So, that sort of determines the size of a cell. But again, this does not mean that all cells are of the same size, that is 10 micrometer in dimension. The largest known cell is that of a egg of an ostrich. So, it is 15 centimeter in width and around 1 centimeter in length. So, 20 centimeter in length. So, it is a very big cell. On the other hand, the smallest cell that is known is around 0.3 micrometers in size. So, you can see that there is a large variation in the size of cells, and it depends completely on the function that cell is going to perform. So, here are some typical examples. So, this is a bacteria, Lactococcus lactis, and this bacteria is used to produce cheese. So, it is used a lot in milk product industry. This is another bacteria. So, this is a bacteria and this is an archaea. So, it forms this colonies like structure, and it is called methanosarcina. This type of archaea, they are found in many places. One of the prominent places is the gut of cattle, and they produce their energy by reducing carbon dioxide and produce methane gas. So, they turn out to be one of the largest producers of methane gas, and methane gas is a greenhouse gas which results in global warming. These cells are found in our blood. So, the red ones are erythrocytes and the white ones are leukocytes. So, this is red blood cell, this is white blood cell. These are false colors, but they have been colored so that we can see them. So, we have already seen these red blood cells, they carry hemoglobin, which is very important for transport of oxygen. We will see about these leukocytes in more details when we talk about our immune system. So, there are different types of leukocytes and they have different functions, and they form a very important part of our immune system. This is a neuronal cell. So, it is a Purkinje neuron. It is found in our brain, and you can see that its structure or morphology is very different from these cells, right. So, this neuron, it has all these branchings and it makes connections. So, this particular neuron can make almost 100,000 different connections with other neurons, right. So, cells, they come in all sorts of shapes and sizes. Now, again, broadly, if we look at cells, which I have already shown you before, they can be either prokaryotes or eukaryotes. So, what does that mean? Prokaryote means these are single celled organisms and these are all bacteria. So, this can be eubacteria or archaea. Eukaryotes, on the other hand, can be single celled or can be multicellular. Pro means before and eu means true. True means something that is present. What is present? Nucleus is present. So, prokaryotes are the organisms which evolved before nucleus. So, they do not have nucleus, and eukaryotes, they have nucleus. So, that is why it is true that the nucleus is present. So, general architecture of these two cell types will be something like this. This is a bacterial cell. So, what we see here is the plasma membrane and there is a cell wall. So, we have this inner plasma membrane. So, if we zoom into this, we have an inner plasma membrane and then we have a cell wall and then we have this periplasmic space, right, and then outer membrane. So, we will talk about this in more details in later lectures. On the other hand, a eukaryotic cell looks more complex. So, it is much bigger in size than a bacterial cell. So, you can see that this is 0.5 micron in scale and this is 1 micron, okay, and this is much bigger. So, you have to actually make it twice the size of this if you put it in this scale, and you will see it is much bigger than a bacterial cell. So, here we see nucleus, we see all these different organelles, right. So, the architecture of a eukaryotic cell is much more complex compared to a bacterial cell. What we see here is the DNA of the bacteria. The DNA of the eukaryote will be present inside the nucleus. So, there are of course some similarities. Both have plasma membrane, they have cytoplasm. So, this part or this part is the cytoplasm. The genetic material is DNA in both cases. They use energy currency. Typical energy currency will be ATP, and they use enzymes and coenzymes to perform various tasks inside the cell to carry out all the biochemical reactions. But then there are of course several differences. One difference is that no membrane enclosed intracellular compartment is present to house the DNA, right. We saw that in the previous slide. In case of eukaryotes, there is a well defined nucleus which houses the genetic material, which is the DNA. Prokaryotes or bacterial cell lack most of the complex membrane bound internal organelles. So, there is no mitochondria, there is no ER, there is no Golgi, but this type of complex membrane bound internal organelles are present in eukaryotic cells. Prokaryotes have a single circular chromosome, whereas eukaryotes have paired chromosomes, and we will see examples that in many cases these are linear chromosomes. So, here the chromosome is a single chromosome, but in this case we have multiple chromosomes. So, the DNA is divided into multiple molecules and these are linear molecules. Okay. Prokaryotes lack histone proteins, eukaryotes have histone proteins. So, I will show you what is a histone protein in the next few slides. And finally, prokaryotes have a peptidoglycan cell wall. Plant and fungal cells have these cellulose and chitin in cell wall, but no such cell walls are present in animals. So, we do not have cell walls, and this is something that becomes very important because we can target this peptidoglycan cell wall to kill pathogenic bacteria, because since we do not have that, any molecule which targets a cell wall will not cause any harm to us. But of course, you have to remember that they will kill the bacteria or the good bacteria which are present in our body. Okay. So, I mentioned that in eukaryotes DNA is present as paired and linear molecules which are called chromosomes. So, this is a eukaryotic cell, this is the nucleus. Inside the nucleus, chromosome will be present like this. So, if we zoom into this, we can see the DNA, but this DNA is present in a highly condensed form like this, and this is something that happens in a particular state of the cell cycle which is called mitosis. So, I will talk about mitosis in more details in the third lecture of this week. If we consider the human genome, then our DNA is divided into 23 fragments, and depending on the size they are numbered as 1, 2, 3, so on and so forth. So, 1 is the largest chromosome and 22 is the smallest chromosome. The last one, the 23rd chromosome, is actually the sex chromosome. So, in all this 1 to 22, they are the same chromosome and they are present in 2 copies. It turns out that one comes from each parent. The sex chromosome are different, one is X, the other one is Y. So, if an individual has both as X chromosomes, that will be a female. If one is X, one is Y, that will be a male. So, I will discuss this in more details in the third lecture. Now, I would like to point out something that is very interesting. So, if you look at this last chromosome, 22nd, which is the smallest one, it has roughly 48 million base pairs, and if we stretch it out like this, then end to end its length will be 1.5 centimeters. So, you can easily calculate that. You know the distance between the base pairs and you multiply that by 48 million. So, you can figure out what will be the length of this chromosome if we stretch it out completely. In mitotic state, this chromosome is compacted like this and its length is 2 micrometer. So, there is a compaction of 10,000 fold. How do cells achieve this huge amount of compaction? So, mitotic state, the compaction is the largest, but even if we consider an interphase, there also the compaction will be 1000 fold that of this length, right. So, how is that achieved? That is achieved by something using the histone proteins. So, first of all, you have to consider this DNA molecule. So, if you remember, DNA has this phosphate backbone which is negatively charged, which means that DNA is a highly negatively charged molecule. So, this molecule, since it is highly negatively charged, it will expand out, right. So, if we try to compress it into a small volume, it will repel and it will expand out. So, first thing that you have to do is neutralize this charge on the DNA, and that is done by a set of proteins called the histone proteins. So, these histone proteins are the surface of these histone proteins will be highly positively charged, and you can imagine, based on what we have learned so far, what type of residues should be present to make it positively charged. So, there has to be basic residues like arginine and lysine and histidine. So, histone proteins actually have a lot of arginine and lysine and histidine on the surface. The DNA is wrapped around the histone proteins like this. So, it turns out that there are 8 molecules of histone proteins which come together to form this cylinder like structure. On that the DNA is wrapped, and this neutralizes the charge. So, this to this, so this whole histone protein and some part of the DNA which is extending out, this is called a nucleosome, and the DNA length that goes from here to here, considering whatever is wrapped here, will be roughly 200 pairs of nucleotide. So, this is a nucleosome, and if you look at the DNA, it looks like a bead on a string. So, we have this long string, and on this you have these nucleosomes which form small bead like structure. Now, this is almost neutralized. So, this can be compacted like this and more proteins come in to form compact structures like this, right. So, normally DNA is present something like that. In case of mitosis, higher order compaction occurs and the DNA becomes really compact. So, this way we can, or cells, pack this highly negatively charged molecule into a very small volume of the nucleus. So, histone proteins, they look something like this. So, you can see it is mostly alpha helical, these are the protein and the DNA is wrapped around the histone protein. So, there are 4 types of histone proteins, H2A, H2B, H3 and H4, and each of these are present twice. So, there are 2 copies of each of these are present, which means that there is a total of 8 proteins and together they form an octamer which forms this cylindrical structure. On that 146 nucleotide wraps around this histone 1.65 times. So, it goes around like this and then some more, right. So, that is 1.65 times and that is 146 nucleotide base pairs. A diploid human cell will typically have 30 million nucleosomes. So, one more thing that I would like to point out is that this, you can see that the histones are highly structured molecule on which the DNA is wrapped around. However, the end terminus of the histone is shown here like these dotted lines. So, these dotted lines indicate that this end terminus, there are several residues, maybe around 10 residues roughly, are highly disordered, which means that they do not form any structure, they do not form any secondary structure and they do not pack against this rest of the protein. So, they are disordered means they can flop around like so. And this disordered part plays a very important role. So, that disordered part is shown here. So, this is the folded histone. So, this is H2A, H2B, H3 and H4 and their N terminus disordered part is just shown here. This N terminus part undergoes covalent modification. So, these are called post-translational modification because after translation this protein is formed, but there are other enzymes which will come and add certain functional groups on the side chain of a protein. For example, P is a phosphorylation. So, a phosphate group is added. We have already seen phosphorylation. We saw that kinase enzymes add phosphorylation. A is acetylation, M is methylation. So, there are again enzymes which will add these modifications on very specific amino acids on these histones and there are enzymes which will remove this. Just like in case of phosphorylation, we saw that kinases will add the phosphate group and phosphatases will remove the phosphate group. Similarly, there are acetylation and deacetylation or methylation and demethylation that are done by different sets of enzymes. And it turns out that a combination of this acetylation and methylation indicate certain signals to the cell. So, one such example is shown here. So, what is the meaning of this? For example, if a methylation happens here, it means that heterochromatin formation is favored, so that whatever part of the DNA is there, it will be tightly wrapped and it will be not exposed to the, let us say, the polymerase, which means that if any gene is present in that part of the DNA, it will not be accessible to the polymerase. So, no mRNA synthesis will happen. So, that is gene silencing. On the other hand, other modifications can indicate gene expression. So, the chromatin will open up and it will be accessible to the polymerases and other proteins. So, this type of modifications can also control expression and repression of genes. Okay. So, as I mentioned that there are 2 types of cells, prokaryotes and eukaryotes, and these are some of this shown here. So, bacterial cell will typically look like this, eukaryotic cell or animal cell will look like this, and a plant cell will look like this. So, these are both eukaryotes. Plant cell will have a cell wall, bacterial cell will have a cell wall, no cell wall in animal cell. So, what is the function of a bacterial cell wall? It lies outside the cell membrane in nearly all bacteria except Mycoplasma, the smallest one that we saw, and some archaebacteria. They have two important functions. First, they maintain the characteristic shape of the cell and they prevent osmotic lysis. So, there will be mismatch of salts and other molecules inside and outside, and that can result in lysis of the cell. So, that type of lysis is prevented mechanically by this bacterial cell wall, and that is something that becomes very important for us. So, if we look at the cell wall, so we zoom into this cell wall of a bacteria, it is formed of sugar and amino acids. Okay, so polymers of sugar and peptides. So, this chain that you see is made up of two sugar molecules. One is N-acetyl glucosamine or glucanac, the other one is N-acetyl muramic acid or mur2AC, right. So, these are present alternately and they are linked by this beta-1,4 glycosidic bonds. So, one chain is here, one chain is here, one chain is here, right. So, you can think of it like a fence where we have these wires, but then you have to connect those wires, right. So, you have to cross-link those wires, and that is done by this polypeptide chain. So, this is a pentaglycine and these are 4 amino acids which are L-alanine, D-glutamic acid, L-lysine and D-alanine, right. So, just see that here D form of the amino acids are used. So, there are many ways, there is this whole biosynthetic pathway by which the cell wall is synthesized. I will focus only on the last part. So, what we have is this polysaccharide chain, and so that is shown here. From there, this is hanging out, and then this is the pentaglycine. This is another chain. In that, we have this 5 amino acids. So, last two are D-alanine and D-alanine. There is this enzyme called transpeptidase, where this glycine will attack it and the terminal D-alanine will go out. So, it goes out, right, and you form this cross-link. So, this cross-linking is formed. Penicillin is an antibiotic which inhibits this transpeptidase. So, we have already seen enzymes, we have seen there are inhibitors which can form covalent linkage with the enzyme. So, penicillin is something which forms a covalent linkage with this enzyme and it will completely block this enzyme. If this enzyme is blocked, which means that no cell wall synthesis can happen. If cell wall synthesis does not happen, the bacteria will undergo lysis, osmotic lysis. So, penicillin can kill bacteria, right, and it is a very good antibiotic because this is not present in our cells, it is only in bacterial cells. But then again, you have to remember that taking too much antibiotic can cause harm because it can kill the good bacteria that is present in your body, for example, in your gut. Okay. So, coming back to this is the prokaryotic cell. We have already seen the features of this and it is quite simple, but the eukaryotic cell is much more complex and it has all these internal organelles. But one important thing that is common for both is the plasma membrane. So, this plasma membrane that we have for the one that encloses the cell. Plasma membrane is formed by a lipid bilayer. So, we have already seen when I discussed membrane proteins. So, this is a lipid molecule and it has this charged head group and this fatty acid side chain, right. So, it forms a bilayer like this on which these membrane proteins are embedded. So, it turns out that there are all these different types of lipids, right. So, you have phosphatidyl ethanolamine, right, phosphatidylserine, choline, right, sphingomycin, and we will see that these are all present in the two bilayers of the lipid and they play very important roles. So, typically the architecture of a lipid will be that we have a hydrophilic or charged head group and a hydrophobic tail. I would like to point out here that this is the typical structure of bacterial and also eukaryotic lipid, but in case of archaea, the lipids are slightly different. So, you can see that the fatty acid side chain here has branches, right. You can also have cyclic molecules in this fatty acid side chains. This is an ester bond. So that connects the fatty acid with the glycerol moiety. In this case, it is an ether bond. Here it is glycerol 3 phosphate, here it is glycerol 1 phosphate. So, there are some major differences between bacterial and archaeal lipids. So, structure wise both look like this. So, this is something that we normally see for a surfactant. So, the soap that you use, you will have a long fatty acid side chain and a polar head group, right. So, there it looks like a cone because there is only one long chain, and when you put these molecules in water, they will form a structure like this, a spherical structure like this, which is called a micelle. So, it is a micelle. But the lipid that we have in our cells, they have two fatty acid chains, and depending on the cis and trans bonds, you can actually have a kink like this, right. So, the shape of this is more cylindrical instead of a cone. So, if you have a cylindrical molecule, if you pack them, you cannot form a circle or a sphere like this. So, in this case, they pack more like this. So, they form a bilayer, right. Now, if you have a bilayer, this is energetically quite stable. However, there is a problem that the edges will be exposed to water. So, one way you can eliminate that is if you fold it into a complete spherical structure, then there are no edges and this becomes energetically favorable. So, that is your cell. So, whatever is inside will be the inside of the cell and outside will be the outside of the cell. So, intracellular and extracellular parts are separated by this lipid bilayer. On this lipid, there are of course proteins, but there is one more interesting and important molecule which is cholesterol. So, this is the structure of cholesterol, and you can see that it has a tiny polar head group which is this OH group, and then the rest is non-polar, right. So, it is not as big as the lipids, but it sort of spans the hydrophobic part of the chain, and the function that this cholesterol performs is it makes this, it stiffens this region. One of the functions that this cholesterol performs is that it stiffens this region. So, this is something that we have seen before. We have the lipid bilayer, on that we have all these proteins which are embedded. In most cases they are helical, you can also have beta barrels. It turns out that the regions where proteins are embedded, we find more cholesterol molecules because they tend to make this region more rigid, and this type of rigid regions are sometimes referred to as lipid rafts. Also, it is important to note that the two leaflets of the lipid bilayer do not have the same composition of lipids. So, the lipids on the outer membrane versus the lipid on the inner side. So, this is the cytosolic part and this is the extracellular part. They will have different composition, and this is also something that is important. We will see when we discuss about cellular apoptosis. So, if we look at the internal structure of bacteria, they are relatively simpler. There you have ribosome, there is a nucleoid region which has the DNA, there are some big vacuoles, and certain bacteria sometimes contain endospores. So, this is the bacterial architecture. It has DNA, normally it is a singular molecule which is circular. E. coli DNA measures around 1400 micrometer in length, and this DNA is coiled extensively. So, you can see it is a very coiled structure, and RNA and other nucleoid proteins are also present, which forms this nucleoid. Apart from this, there can be other smaller circular DNA present, which are called plasmids. Now, let us look at the ultrastructure of eukaryotic cell. So, a typical eukaryotic cell will look something like this, where we will have a nucleus. Attached to the nucleus, we will have the endoplasmic reticulum, then there will be Golgi bodies, there is mitochondria, and then lysosomes, peroxisomes, and other molecules, other membrane bound structures. So, this table is something that summarizes all the functions of these different membrane bound structures. So, you can pause this and go through this table. This table shows the relative volume of these different organelles that are present in a cell. So, in this case it is given for a typical liver cell. So, volume wise cytosol has the largest volume. So, it occupies more than half of the cellular volume. Then the next big organelle is mitochondria. So, again mitochondria is not one, there are multiple mitochondria is present, and if you consider their total volume, that will be more than 20% or 1/5 of the cellular volume. Then rough ER, then Golgi bodies, and these are much smaller. So, nucleus occupies only 6% of the total cellular volume. So, you can imagine the level of compaction that is needed to pack the DNA inside this nucleus. So, quickly I will go through these different cellular compartments. So, cytosol is this rest of the cell, right, inside the plasma membrane. So, it is a gelatin like fluid that lies inside the cell membrane. Its function is it contains all the salts, minerals and organic acids, and this is the place where a lot of biochemical reactions happen. So, it surrounds all these organelles. Nucleus is here and it is the one which contains the genomic material. So, this is where DNA replication and synthesis of mRNA takes place or synthesis of RNA takes place. So, this is a zoomed in structure of the nucleus. So, this is an electron microscope image. In the next lecture, I will discuss in more details about this type of images. So, if you look at this, there is a nuclear envelope that is present on the nucleus, and that looks more like this. So, there is this nuclear envelope and there are pores which are called the nuclear pores. Okay. So, a nuclear pores are these small structures like this which allow passage of molecules through these pores. So, there are certain proteins which can pass through this only if they have a signaling sequence. So, that signaling sequence will determine whether a protein will remain inside the nucleus or whether it will be transported out, or if something is outside, whether it will be transported in. Now, attached to the nucleus is this endoplasmic reticulum lumen. So, this is endoplasmic reticulum which is connected to the nucleus, and you can see all these dots, these are ribosomes which are embedded on the endoplasmic reticulum. So, when this happens, it is called rough endoplasmic reticulum. So, ribosomes are attached on the membrane, and if there are no ribosomes, it will be called a smooth endoplasmic reticulum. So, its function is intracellular, it is an intracellular highway, right. So, it forms a path along which molecules move from one part of the cell to another part. So, and since you have this ribosome attached, you can imagine that protein synthesis also occurs here. So, again these are the electron micrograph of the rough ER and the smooth ER. Next, after this endoplasmic reticulum, what we have is the Golgi apparatus or the Golgi complex. So, this Golgi complex, they form this stacked flat sac like structure, and what they do is they receive the proteins from the rough ER and they distribute into smaller organelles which get transported out into vesicles. So, you can see a vesicle which is budding here. So, they get transported out. Now, if there are proteins which have to be secreted, they will be secreted out. If there are proteins which have to be delivered to some other organelle, they will be delivered, right. So, processing, packaging and shipping of these molecules are done by the Golgi apparatus. Then there are mitochondria and chloroplast. So, I am not going to discuss much about this because I will talk about them in more details when I talk about respiration and photosynthesis. So, these are the energy producing or the powerhouse of the cell. You can also think of them as the kitchen, right. So, this is where energy is produced, and I will talk about these organelles in more details in a week's lecture. Another type of organelle that is present is called lysosome. So, this you can think of as the garbage disposal of the cell, right. So, lysosome, it has more acidic pH. So, cytosol has a pH of 7.2, lysosome will have a pH of 5, right. And here all these macromolecules which have performed their task or they have damaged or they are not required, they will be taken here and they will be shredded into their monomeric components. So, proteins will be degraded into amino acids, nucleic acid will be degraded into nucleotides, and they will be recycled so that new DNA or RNA or protein can be synthesized. And we have already talked about it when I discussed membrane protein, that we talked about an example of hydrogen pump, right, where ATP is used to pump in hydrogen, and that is why the pH of the lysosome is maintained in an acidic pH, which is pH 5. So, for this lecture you can follow these books, especially Molecular Biology of Cell by Alberts and Molecular Cell Biology by Lodish. Thank you.